we've heard that innovation is teamwork, and it's also a flow of things going from innovative research and great ideas. So I would like to welcome Dr. Barbara di Ventura from the University of Freiburg, who will be talking about Inteins as molecular tools for synthetic biology. Thank you very much. So I think, can I take this microphone? Yeah, if you want to move, yes. Yeah, to move, exactly. I like to move. Maybe it's working. You can start here and I try to figure it out.
So I'm very excited to be here. Thanks to Ariel for having invited me. So from the neighboring country of Germany, actually originally I'm Italian, I guess my name tells it. So we are gonna go into core molecular biology / synthetic biology now for this talk. And I'm going to present to you some very interesting proteins called inteins that perhaps can make this jump from just basic science to innovation. So actually maybe I have to do it here, yes. So in synthetic biology, we of course like all the molecules that the cell is doing, but proteins are for us similarly interesting because they are the workhorse of the cell, they perform functions. So if we can take hold on the proteins and tell them what to do, we can of course create constructs that will act in a certain way. So for what was said in the talk before, for instance, for biomedical purposes or for bioremediation, considering the situation that we currently have with the environment, this is also very important. So one of the features of proteins that we really like is the fact that they are made of independently folding domains. And these domains have the ability to also work, so to perform the function also when they are extracted from the context of the full protein and expressed in the cells separately or even in vitro. And that's why you sometimes have heard synthetic biology talk about playing with Lego or so, because we can take these parts and then put them together in new ways to create proteins with functions that did not necessarily exist in nature before. But in order to do this, we need to take these parts of the proteins and then stitch them together in different ways. So scientists have done that also in contexts such as creating new synthetic transcription factors. So what are transcription factors? These are proteins that activate gene expression. And because, of course, the activation of gene expression can lead to a lot of functions in the cell, this is an important process to control for synthetic biology reasons. So how is this typically done? Because of this power of proteins to be able to be separated in the different domains, we can take the domain that binds to the DNA and the domain that binds to the other proteins in the cell that activate transcription and express them separately. And as far as we have a way to bring these two parts, again in physical proximity, we can reconstitute a functional transcription factor. And this is actually what lies at the core of the so-called yeast-to-hybrid technology that you might have heard during your projects or your studies, because it actually is a way for you to know if protein A and B are interacting with each other. Because then you fuse these two domains to A and B, and if A and B interact with each other, they will bring these two domains of the transcription factor in physical proximity, and you will get, for instance, the expression of a reporter. So this happens when we have, let's say, we are really talking about taking one domain and another domain and bring them together. But sometimes we might want to split a protein, so divide a protein into parts, not necessarily according to these functional domains, but somewhere else. And it is possible to do this, so to take a protein and take two parts out of it, and then if we can bring them again close to each other physically, and of course if these two parts are stable and they can regain the proper structure, the 3D structure, we can gain again the function of this protein by simply bringing these two parts in close proximity. And this is what happens with the fluorescent protein GFP very, very often. This is again a very used tool in molecular biology or synthetic biology because it allows you to have a very easy readout. Fluorescence is reconstituted if you fuse these two parts of GFP to the protein A and protein B, and you want to know if these two are coming together. So until this point, we're talking about methods to rather visualize in an easy way whether A and B are working together, are binding to each other. But I want to introduce to you these new proteins, not new, of course, so they came out of evolution, so they're definitely very old, but so they are gaining momentum in the moment in synthetic biology because of the many possibilities that allow you to do in a synthetic biology context. And these are actually inteins. So what are inteins? You might have heard about introns, so at the level of the mRNA, and inteins are also similar in that sense because they are sequences which are hosted within host proteins, but they splice themselves, so they eliminate themselves out of these host proteins. And in doing this, though, what they do, which is very interesting if you want, is that they create again a peptide bond between the flanking regions. So in this case, you might start with a protein that doesn't have a function. What is the pointer? Do I have the pointer? It doesn't work, so I don't need to point. I can do it with my body. So if I am the intein, and these are the two extain sequences, so we call extain whatever is flanking the intein, and then I am expelling myself, so I'm splicing myself out, then I will create a peptide bond between these two flanking regions. And now this is a new protein, really with a peptide bond in between, which is indistinguishable from the peptide bond that was made or would have been made by the ribosome. So this is, of course, as you might already imagine, a very powerful tool because it works at the post-transcriptional level, at the post-translational level. So the protein is already there, and you can change it. So it can act very quickly compared to controlling gene expression. So I will not bore you with the details. Probably here is not the right audience. What do you really need for these inteins to work? Of course, there are some features that you need to preserve. Some residues that are very important, amino acids that are conserved and are very important for the chemistry of this reaction to occur. So at the moment, I will skip it. But what you need to know is that it is a self-catalyzed reaction, so you don't really need any other cofactor or any other enzyme. So this is a depiction of what I told you before. So what we can do is that then we have these inteins, so these inteins come in nature in different flavors. They can be contiguous, meaning they are a single polypeptide, or they can be expressed in the cell as two different fragments that need to come back together, make complex first, and then do the splicing reaction. So these are called split inteins. And these split inteins allow you to do a bit more than maybe the contiguous intein, because now you can have two separate polypeptides, two separate proteins, and when the intein fragments interact with each other, they splice and they make now a new protein that is made of the whatever was upstream of what we call the N intein and whatever was downstream of the C intein. So this is now a protein that is really up to you to make, so it gives you a huge power, if you want, for any application you have in mind. So we used it many years ago with an iGEM team. So I don't know if in this audience people know about iGEM. So iGEM is actually probably the closest to what is happening here today, right? So it's really taking students, asking them to perform a project during the summer, and then come back, go to a big event where teams around the world gather all together, and they present their project, which are very often super applied because they are in the context of synthetic biology. So in 2018, we were working with a team. No, actually, it was not 18, it was 2014, so longer than this. We were working with this idea of using the inteins to circularize proteins. So what is this? So typically, proteins have a beginning and have an end. So we call the N terminus and the C terminus. And these termini are very often decided which proteases can attack the protein, so they can lead to instability. So the protein can be degraded. If we now bring them together, so we make an unnatural linkage between these termini, the proteins will have been shown by several researchers before us that they become more resistant to protease degradation, they are less prone to aggregate, and they have a higher thermal stability. So we thought, can we use this to make proteins which are more heat resistant? And this, again, is a step towards innovation, if you want. So we never brought it to that level. I guess today we will hear a lot about what it takes from going from basic science to real application, real innovation. So we reached the point of making some proof of concept to show that you can then use this technique of circularization to make a protein, an enzyme that is used in the industry to make it more resistant to all of these things I told you before. And this could potentially be used in a plant later on without having to decrease the temperature or use other harsh chemicals, because these proteins would be resistant to this. And how it works is we use the inteins, and you need to put them in a special location, so between the N terminus and the C terminus, and when the protein will be, or the inteins will be splicing, they will create this peptide bond and circularize the protein. So then actually we went farther, and we also decided to use these inteins for another, to create another tool that can be very useful in synthetic biology in many ways, because we, when we work, for instance, with bacteria, we very often work with at least two plasmids. So plasmids is this circular DNA that can be transformed into the cells and brings in the genetic information we want the bacteria to have in order to produce something like a chemical for us. And so we rely on these two plasmids, and typically the way in which we have to keep this, or the trick that we use to ensure that the cells keep this plasmid and don't kick it out, is we use antibiotics. And these plasmids give resistance to the antibiotics, so the cells that contain the plasmid survive in an environment with the antibiotics. And typically when we want to have two plasmids, we have two antibiotics. And despite the fact that this is something that is used a lot since many, many decades, still we thought it would be more convenient if we could decrease, for instance, this amount of antibiotic from 2 to 1, so to allow people to be able to screen for cells that contain two plasmids using a single antibiotic. So how did we do this? The idea of my student back then was to, again, go back to splicing an enzyme, the enzyme that confers resistance to the antibiotic, into two parts. And of course, each part, importantly, should not have the function, right? So each fragment of the enzyme should be inactive, but only when the two parts come together, get spliced together, you get the function again. And therefore, we created two cassettes. In one plasmid, we express only one part of the enzyme fused to an intein fragment, and in the other plasmid, the corresponding intein, that the partner, if you want, so the other fragment of the intein and the second part of the enzyme. And we thought when the cells contain both plasmids, they will express these two parts of the enzyme, the splicing should occur, and the cells should survive. The challenge was how to actually, this is what I told you already. So the challenge was how to really split the enzymes, because this is not really a banal thing to do, to take a protein and simply say, okay, I cut it here, express these two parts, and these two parts should fold properly and still have the ability to then, first of all, allow the intein splicing. And so many things have to come to place. So again, I will not go too much into the details, also because of time constraints, but you can ask me, please, later on. So the idea that we had was to look a bit the structural features of the proteins and also at protein flexibility, looking at somehow the parts of the proteins in the loops, if you want, for instance, so where there is less conservation and where there is more accessibility of these sites, because we know that if a site is conserved, it means it's very, very important for the structure, for the function, and you better not touch those. While if you look at less conserved parts, it means that these parts can change and it won't affect too much the protein. So that's where we looked for the split sites, and we created this tool that we called Simple for split intein-mediated selection of cells containing two plasmids. And this is a tool that now allows you to use a single antibiotic to select bacteria, at this stage is still bacteria, that contain two plasmid. And we did it with a lot of enzymes, because people don't use only, say, ampicillin, but they use kanamycin, chloramphenicol, puromycin, hygromycin, so many different ones, so that we create a toolbox or a toolkit with many different flavors, if you want, that you can actually get from Agin, which is a public repository where you can get your things. So you don't have to pay us if you want, so you actually pay them just the maintenance of this repository and the shipping of the constructs, but we don't make the money out of it. So maybe that's a failure in innovation in this context. So we just give it for free to you, to some degree. Okay, and what am I doing? Yeah, I'm not skipping. Okay, so very briefly, so what we wanted to show with this is that now if you use this concept of reducing the amount of antibiotic you need to use to grow these bacterial cells, you might have cells which are happier, healthier, and can produce more of a certain good, if you want. So this is metabolic engineering, is done a lot in synthetic biology. That is to tell the bacteria to produce something that you want in large quantities, and people struggle a lot to achieve these large quantities. So whatever trick you can use to make the cells happier to produce larger quantity, that is welcome. And so we thought maybe in this case, if we take already a pathway that requires the expression of 6 enzymes, 6 is just a number, so it could be any other thing, that is that they are indeed encoded into 2 different plasmids, and if we now change these plasmids into the version of the simple version, so we can now use 2 antibiotics, maybe we can, antibiotics, and maybe we can get higher level of the product, and this is what we observe. So it's not unbelievably a lot, I have to say. So of course there are methods that can show a much higher increase in the yield of a product that is being made, but nonetheless, I mean, this at least showed us as a very, very preliminary proof of principle that this could be a viable solution. And up to this is really only still in bacteria. But actually then we moved on, and we know that in the field of mammalian synthetic biology, so people using mammalian cells, actually when you speak to the researchers, they say there is one antibiotic that works very well, which is puromycin, and everything else is already way more complicated. So now for people wanting to create more complex synthetic networks, synthetic circuits, having the possibility to use only puromycin and allowing the cells to, or actually pushing the cells to obtain 2 different vectors, that would be very important. And that's why we did this, that we went to the version of simple now for lentiviral constructs. And some of you that are familiar with lentiviral constructs, they can say, oh, but I can always express fluorescent proteins and then sort with FACS the cells that contain these 2 markers, and I don't need this. But not all the times you want to spend your fluorescent channels to just sort your population. So sometimes you want to do experiments then with this, and you want to rather use other ways to ensure this population to be pure. So that's, in this case, that's what you can do. You can impose puromycin resistance and then select the cells that will be 100% pure for the 2 lentiviruses. So you, of course, have 2 separate lentiviruses. You mix them together. You infect the cells with these 2, with a population of the 2. You make them undergo, this puromycin treatment, and then you select. And at the end, you will have a double positive population. And this is exactly what we show here, right? That if you get the untransfused, so these are, for you who are not familiar with this histogram, so this is a flow cytometry, so where we are looking at fluorescence, and we're trying to find the cells that are positive. So these are the ones that are shifted towards for you is the right side. So at the beginning you see very, very little cells are fluorescent. This is some spurious production there. But when we transduce with our virus, without the selection, so you get some cells that are pure, but when you have you made cells undergo this puromycin selection, then you get practically 100% of the population that will contain both of the proteins that you're looking for. And maybe I skipped this, is just to say, okay, whatever we were producing was functional, so it was not just some random stuff that we were making. In the last, I don't know what's the time that I have? 5 minutes, oh, so I have to rush like always. I always speak too much, so okay, I have to be faster. So these inteins, right, I told you before, they're coming either as a contiguous or as a split intein. And you can ask yourself, maybe sometimes you want to have an intein which is split at a different side, yeah? And this is actually has happened in the past. So people wanted to change the location at which the intein would be expressed as 2 different parts. And at the moment, this has always been done in a heuristic way. So people just try, okay, I split it here, let me test. And we decided to create a machine learning algorithm. So it's a web server that all of you can use, once again, for free, to test whether you can now, so to predict, if you want, where an intent that was never split before or that was split before but done heuristically can be split and be active. And this web server is called Inteins, so with the only difference that the A is now a end, if you want. So I will probably skip all of the details about how we did it. Sorry, guys, so you can ask me, but I just want to say it works very well. So this is now a Gaussian naive Bayes type of algorithm, and it can perform quite well both on the training data set. So the training data set, you might have heard this machine learning. Everybody speaks about AI and machine learning. So you need a very good data set. And the data set that we had from the literature that we tried to start with was not good enough. Why not good enough? Because it was very biased towards the sites that are working, because people that publish papers, they publish the success story. So they publish when they have split an intent and the intent was functional. But in order to create a model, you need both the good and the bad sites, also the ones that do not splice, because then you have an unbiased way to predict and find the features that are important. So we created our own data set, which was a lot of work for my master's student back then, but he decided to go for it, and the algorithm works quite well, so it has an accuracy of 0.78, precision of 0.78, and recall and the Matthew correlation coefficient of 0.56, and it, importantly, which is a very important point, it can also predict whether the fragments, the two intent fragments, will associate on their own or whether they will need a push, if you want, and this is something that one can use to create conditional intent. So this means that instead of splicing naturally, you need to push the splicing to occur, so meaning that you can take, again, the upper hand and you can decide whether the splicing is occurring or not by using, for instance, inducible heterodimers. So this can be via drug like rapamycin or rapalogs, or light, which is something that in my lab we do a lot, so in optogenetics. So this we think is very useful because intents are being used in the context of conditional intents, and if you want to use another intent and split it at a different site, now you have an algorithm that will help you predict. And so this is how the performance is on a unbiased now data set, so the literature data set that was not used for predicting the model. So this is completely independent, and it works still very well. What did we do with it? We tried to control the Cre recombinase. The Cre recombinase is used again a lot in general molecular biology, even mouse genetics a lot, because it is a recombinase, so changes the DNA and allows you, for instance, to flip a gene sequence, so you can have a gene all of a sudden being made if you have the recombinase, or you can eliminate a stop cassette, which also allows you to have a transcription of the downstream gene. And Cre recombinase practically almost, every half a year there is a paper that is appearing, so people try to control it a lot because it's super important technologically. So we try to use now our algorithm to do the same, to split the Cre, and this is not something that we really created, right? So split Cre is present in the literature since long, but we now use our algorithm to predict where we could split the intane to then make it conditional. So by putting two components, what you read there, I lead SSPB, these are just two proteins that bind each other when there is blue light on the cells, okay? So like this, we can control the splicing reaction with blue light. And here is the first data that we have acquired, and it is working. So the blue bar is the activity of the Cre, so how much reporter has been made when we have blue light, and in the gray is what happened in the dark. And what you need to understand is that at least when you work with optogenetics, which is using light to control reactions or in the cells, it's always a trade-off between lowering the activity in the dark, so you want to not have the things happen before you give the light, and also lowering, unfortunately, what happens in the light. So it's the wish would be no activity in the dark and a huge activity in the light, but we are constantly fighting between these two. And so here you see, if we push lower the gray bar, then also the blue bar comes a little bit low. So we have to come up with strategies to maybe allow us to do this better. Okay, so we are at the take-home messages. I hope you are now all convinced that Inteins are super cool. Actually, there is, of course, also a very famous French researcher who was working with Inteins, and I'm so bad with names. I'm very sorry. Probably he's even sitting in the audience, and I apologize if I forgot your name. But so he's also using Inteins in a very cool way to, I think, reconstitute toxins or something like this. Really nice stuff. And you can use circular protein. So if you want to go in innovation and you want to have a protein that is resistant to a lot of stuff, don't forget that circularization could be useful for you, and you can use the Inteins for that. Well, we can, in this paper, we really show that we can use a rational approach. So there was not trial and error. We really sat on the computer and decided, okay, that's where we can split the enzyme. So this is not the Intein, but the enzyme. And it worked 100% of the times. But I think that this is due to the specific type of assay that we're doing. And if you have questions, come to me, and I will explain to you what I mean. And okay, simple is on Agin, you can use it. So if you feel that you have the need to decrease the number of antibiotics and nonetheless expand the amount of circuits that you give to your cells, so the number of plasmid or lentiviral plasmid, then you can use simple. Yeah, and could be interesting maybe for metabolic projects in bacteria. And Intein is a web server that would help you predicting, I would say with high confidence, whether an Intein split at that side will be functional or not, and if this functionality will depend on heterodimerization system that you put on them, which I think is very important. And in fact, you could use it to create novel conditional Inteins. Yeah, controlling CRE is quite challenging, and we can talk about it if you want later. I want to thank, of course, my lab. They do the work, so I think unfortunately at this stage we just sit and write grants. So we don't do the work ourselves, so the students do it. So without them, practically there is no science. So I thank them, of course, especially the members that have been specifically involved in these various projects. I talked to you about our collaborators in Freiburg. They work with the T cells in the context of the Intein project, and the two researchers in Stuttgart that gave us the ability to test this metabolic pathway in bacteria. Carsten Voigt, who helps us with the web server maintenance, and of course all the funding bodies, and you for your attention. And I hope you can ask me a question if you want. So I hope I didn't go too fast in explaining all of this to you.
Thank you very much for this wonderful presentation and illustrating all the breadth of possibilities of Inteins, and also showing us how they can be used in mammalian cells in synthetic biology. Are there some questions, maybe one or two questions? Yes, over there, you can use your microphone. It's working. You press the icon just under the microphone, the little speaking.
Right. Hi, Barbara. Excellent talk. Thank you very much. I want to know, what is the number of pairs of known orthogonal Inteins?
Few really orthogonal. So we work, there is one pair that has been demonstrated to be orthogonal, GP41-1 and NRDJ1. So these two have been used and they are orthogonal. GP41-1 and MPU-DNAE, they actually are not really orthogonal. So we didn't know so much about it. We did some experiments in the lab, and then all of a sudden we saw cross-reactivity, and we were a little bit surprised. Of course, there are several other Inteins out there, and I'm not so sure whether they have been really tested. So I think at least two and maybe even three. So there was one paper where I think they managed to have even three. I forgot which is the other one. Maybe it was DNAE, SPB, I don't know. But these two, I tell you, they are definitely orthogonal to each other, GP41-1 and NRDJ1.
Other questions?
Very quick one. So, hi, Laura. Thanks for a lovely talk. So we often split proteins into two. Did you ever try to split them into three or four?
Yes. So the Intein itself, we split it into three parts. It works, but has a very low efficiency. So we have a paper that kind of remained on bioRxiv, and I don't think we will push it any further than that for various reasons. But so you can split the Intein even into three parts, and why was that super interesting? Because if we split it into 3 parts and we can control only the middle part, then you have a reconstitution where your control elements are completely out of your final product. That's why we were super excited about it, and we can have a little bit of activity, but it's very low. Probably by pushing with some evolution, right? In lab evolution, maybe you can increase the activity there. And GFP, I think, has been also split in 3. So some proteins have been split in 3 parts. So
and for the antibiotic-resistant genes, did you try?
We did not try to split in 3. Are you interested in that? We can talk.
All right, thank you very much for answering these questions.